just now, has been in my group for the last 3.5 years, and joined my group about a little less than halfway through her PhD, and wanted a new environment. And I have to admit some fault. When you first joined the group, I had you suggested some projects which didn't end up working out, which I hope didn't send you farther behind. But then once we found this area of swallowing therapy in sports medicine and dysphagia, and the construction of devices, the things really started to pick up. So I don't think you'll be able to tell from the presentation today, but this is about 2.5 years of work here, and it looks like a lot more than that. So B was very effective in corralling all of these various different expertise of all of our collaborators, including kinesthesiologists, sports medicine people, electrical engineers, machine learning experts, chemical engineers, and nano engineers, and really was able to be like a project manager at the same time as being a scholar. And I think that that's a skill that not many people develop as they go through grad school. So also B has been an absolute pleasure to have in the lab, has been a real leader, especially with regard to safety responsibilities. So leading our efforts and having almost 0 violations in any of our municipal and campus level safety audits, and has a job in San Diego, so she can continue to be an engineer and a scholar and a triathlete in the most triathletogenic place in the country. And it is my pleasure to welcome her to the stage to talk about her PhD research.
I'm just going to check if Professor Chibukchu is also here before. He's okay. And Professor Nig is also here. Okay. Yeah. Okay. Everything sounds good, Rory?
Yeah.
All right. Okay. Good morning, everyone. Thank you so much for being here at my thesis defense. Again, thank you, Darren, for the kind introduction. Today, I will talk about my thesis defense that's titled Epidermal Sensors for Monitoring Liquid Swallowing Behavior. So you're going to ask, why monitor swallowing behavior? And the first reason I will give you is dysphagia. Dysphagia is a condition defined as any obstruction to normal swallowing. And the causes vary by a lot. It could be neurological. If you have Parkinson's disease or stroke or dementia, these conditions tend to cause dysphagia as a side effect as well. Head and neck cancer survivors, so after they get treatment, the radiation therapy, the fibrosis that forms around the neck usually obstructs the normal swallowing behavior, and it gets worse over time. And also immunological, so it could be any inflammatory myopathies that happen could also cause dysphagia. And other than these 3 causes I gave you, personally, my grandparents had dysphagia, and at the time I did not know that it was called dysphagia, but a lot of the elderly do have swallowing disorders that happen over time, and sometimes this can cause aspiration pneumonia, malnutrition, even anxiety over not being able to eat and drink as comfortably as they used to do. And so these cases actually do hurt people a lot in their day-to-day life. And another thing that dysphagia could cause is dehydration, and also gives another second reason for us to monitor swallowing behavior. Dehydration is also defined as dramatic decrease in total body water content. That's a very general term, and the causes of it is a lot more varying than dysphagia. It could be that you have fever, you have diarrhea. These can cause dehydration, as you may know. Also working in hot conditions, or exercising in very hot conditions. So excessive sweating and heating can cause dehydration as well. And the things that if you go too much into the dehydration, or if you do not get dehydrated or seek medical treatment, it could go all the way up to heat stroke, urinary and kidney problems, which are quite painful, even seizures, and hypovolemic shock as well. So these are pretty serious cases that dehydration could cause if you're not noticing that you're not drinking enough, or maybe it's too late for you to realize by the time all these things happen. So before we dive into swallowing and how we can monitor it, we do need to understand the stages of swallowing. There has been extensive research done in describing how the swallowing happens, and I'm going to introduce you the main one, which is the called 4-stage model for liquid or solid bolus swallowing. The first stage is called oral preparatory stage, meaning the bolus, which is the thing that you ingest into your mouth, is held between the anterior surface of the tongue and the hard palate, as you see up here. So the bolus is this light gray thing that is inside your mouth waiting to be swallowed. So that's the first stage. The second stage is called oral propulsive. So the bolus is propelled backwards into the pharynx area. And in this scenario, the oral cavity and the tongue muscles do push this bolus backwards. The next stage is the pharyngeal stage, which means now the bolus is in the pharynx area. And in order for this to happen, this tiny little thing called epiglottis and larynx meet each other to protect your airway, so that the bolus or liquid does not go into your airway. Instead, it goes back into your esophagus, into your stomach. And that brings us to the last stage, which is the esophageal stage. And at this point, the swallow is complete, meaning that the peristaltic wave motion of your esophagus carries bolus all the way to your stomach. So in this work, we will really focus on these two stages. The sensors that we will use, or the methods that are being used to monitor swallowing, look specifically in this phase of a 4-stage model to be able to diagnose any sort of disorder that happens. And I will introduce you the gold standard of this, because we need to understand what's been out there. So the gold standard for this is called video fluoroscopy, commonly known as the modified barium swallow, or MBS exam. It is essentially a video recording from the side under continuous X-ray radiation, and the bolus that is given for participants or the patients to drink is impregnated with barium. Or barium, am I saying that wrong? Barium, I will click on here and we can watch in the video. So for the normal swallow, you're going to see the patient take the bolus, and it goes down quite easy and fast, not going into any other larynx region. But if you look at the abnormal swallow, the bolus gets stuck in locations that it shouldn't, and it doesn't only go into esophagus, it also goes into the larynx, which makes you cough. And this is what essentially causes aspiration pneumonia if you're not paying attention. So there are a few disadvantages to this video fluoroscopy, even though it is the gold standard, and it's been used to diagnose dysphagia currently in clinics. The first one is, as you can imagine, is the exposure to radiation. So you are getting an X-ray through you in order to get these videos. And if you need to be constantly monitored, if you're in risk of developing dysphagia, you have to get exposed to this radiation every time you go to a clinic. Second thing is it's expensive. You're going to a clinic, you need trained personnel to be doing these video fluoroscopy for you. And it's only available, like I said, in clinic. You cannot get this done at home. You cannot be doing monitoring yourself at home every week, every month. You do have to go in, going to get this procedure done. So this is why we need to think about how can we make a sensor system or monitoring system that we can very easily use. It's non-obtrusive. It is not going to cost a lot of money to the patients, and it doesn't require trained personnel to work. And here comes the epidermal sensors, especially for swallowing detection. There has been a lot of research done on using wearable sensors on skin to be able to measure swallowing behavior. And I'm going to introduce a few different one of them. So the first one is surface electromyography based sensors. Surface electromyography, or I will refer to as SEMG, measures the voltage difference between the two electrodes that are placed on a muscle. So it measures the muscle activity, muscle activation, and measures that action potential that travels from the nerve cells all the way to the muscles to induce that contraction. And a lot of people used for SEMG thin metal films, especially gold, as the electrode, and usually need 2 working electrodes and 1 reference electrode placed somewhere else other than the muscle. And there are 2 studies that have done this where they have wearable sensors in a thin film with about 200 nanometers to 300 nanometers gold, thick gold. The next one is pressure-based sensors. So pressure-based sensors are essentially type of sensors that when a compression on the sensor is applied normal to the material, it induces a change in capacitance, and we measure that. And for this reason, people have come with very novel materials. So for example, this group from Nana et al. Have used aluminum nitride as the active material to be measuring the pressure. The other one was graphene impregnated PDMS sponge. So PDMS is an elastomer, and they were able to put graphene, and when you apply a pressure or force, you do get a change in capacitance in the material, and thus see signals from a swallow. The third one is acoustic based. So people actually have tried to listen to the sound of swallowing. And that sound that I'm talking about is the mechanoacoustic waves that are generated essentially from all of your organs. They do work at a certain frequency, and you can pick up those sound waves when you listen carefully. So a couple of the novel materials people use is laser induced graphene. This was actually really cool when I found out. They use a polyimide or Kapton tape, and they shoot a laser to it, and you can form graphene layer on top of it. And they use that as detection of the sound waves. The other one, which is not really a novel material, but used commercially. So people have bought accelerometers and microphones, just put it on people's throat and listen to the sounds, and try to understand more of the swallowing behavior. And lastly, I will introduce the strain-based sensors. Strain-based sensors, similar to pressure-based, slightly different. So when you deform these sensors, you can actually get a change in electrical resistance across the film. And what you're measuring is like the strain change on your skin when you put the sensor over the skin. And people have used actually a lot of different materials for this one. There is quite big of literature on this. So for example, Raw et al. Used single-walled carbon nanotubes. A lot of people used silver nanowires, and they usually embed this in an elastomer like PDMS, or combine it with conductive polymers like polyurethane and PDOT-PSS. Another group used fibers. So they use polyurethane fibers and coat them with graphene nanoplatelets to make it conductive, and use that as the strain gauge to be put on the throat. So after these, after I introduce these, I will actually introduce what we have done in our lab previously. So this is before my time. Ramirez et al., a grad student from our lab previously, have used these sensors as strain to be used on the skin to measure different stuff. So I will first introduce what we have done in our lab. We have found that metallic nano-islands, when they're evaporated on a monolayer of graphene, less than 10 nanometer thickness, they actually perform really good as a strain sensor. And they're able to detect strains all the way down to 1 parts per million, or 0.0001% strain. This is an SEM image or electron microscopy image of how these nano-islands look like. So the dark in the background is the graphene, which you're not able to see. And this light gray area is the gold nano-islands evaporated onto the graphene. And this is when it's evaporated at an 8 nanometer thickness of gold. We need to be able to discuss numbers. How do we know this is a good strain sensor? So there is this term called gauge factor, which describes the change in resistance across the strain film over strain that is applied onto the sensor. So we usually make a step edge and put the sensor in the middle of it. So this could be a 1 micrometer or 10 micrometer step. 1 micrometer step is how you can create a 1 part per million strain onto the sensor. So by keeping one side constant and bending the other side, you can induce that bending strain on the film and get a resistance change across the film and calculate your number gauge factor. So we have done this in lab before, or it has been done in our lab before, and it was found that these sensors, strain sensors, were really high in sensitivity, especially in the low strain regime. And I'm talking like cell contractility kind of forces I'm talking about. And the gauge factors were over 9, over 10, which is a lot higher than what all the other strain based sensors that were made with novel materials. The second one is that it had a really high resolution, meaning that it was able to detect very small strains like I described above. And thirdly, this is actually really important, the film that we are using, normally the metal films, when you heat them up or when there is a temperature gradient in the film, the resistance, baseline resistance does change over time, even though you are not moving or applying any strain. So we don't really want that. We want that resistance, baseline resistance, to be constant, even though when there's no strain applied. And so the 8 nanometer gold was found to have a near zero temperature coefficient, meaning that it does not change its resistance, even the temperature is being risen. And you see in this plot over here that was published before, graphene on gold for 8 nanometer had very little change in resistance, which is what we were hoping to see. So the sensors that were used in our lab previously, like I said, it was used for detection of swallow. So again, Julian have done this before. He used palladium nano-islands on graphene, and he has put this thin film onto polyimide or Kapton tape, as maybe you have seen them, the orange tapes before. And he put that onto people's neck, and he was able to detect swallow on people. And he also has done this with people who were suffering from dysphagia and people who were healthy and not having any dysphasic symptoms. And there were good things that came out of this study. For example, it was shown that people who suffer from dysphagia take longer time to swallow because it is a lot harder and it takes multiple swallows to get that bolus in. Secondly, he has done this sensor with a little bit of more adjustment. He had added a conductive polymer called PSS to be able to have the sensor work for larger strain range. So I'm talking about, like, if there is any kind of bending that is larger than 10% strain, you do need to have a secondary layer on top to let electrons travel through if that thin graphene film does crack. So he has done that and actually showed that you can detect breathing when the sensor is placed on the chest, and also respiration or heartbeats. So today I will actually combine these two studies and talk about my first project, which is predicting different swallowed liquid volumes using machine learning. And second part is going to be improving a little bit upon that. So detecting swallow in dynamic conditions and not just stationary seated. And the last part is improving a little bit more on that, adding more strain sensors per patch to be able to get better algorithm predictions. So starting with the chapter 1, the first goal is that, or the question we have asked is that, is it possible to even detect small differences between different swallowed liquid volumes? We have done that with different boluses where the person drinks liquid, yogurt, or cracker, when I say we, previously in our lab, but it wasn't done between small differences in volume. So what I have done or designed the experiment to be is we have cohort experiments. We get people in, we have them seated with sensors on their throat. We give them different volumes of water. And the only difference is there's 5 ml increment difference between each. After the signal collection, we do feature extraction from that and then use machine learning algorithm to be able to predict those volumes, what they have drunk, what they have swallowed. And before I dive into the details of it, I do want to show how we make these strain sensors because it looks very simple. However, it takes multiple steps. We start with graphene on copper. So we buy graphene as a monolayer grown on copper commercially. We evaporate gold nanoislands on top of it in lab. Then we use a support layer called polymethyl methacrylate, PMMA. And we also use a tattoo paper for ease of transfer the sensor onto skin as the base layer. And the next step includes transfer of this thin film that we made with graphene and gold nano-islands onto this tattoo paper. And we, last step, spray coat PDOT-PSS to give electrons a second layer to move around. After adding our copper wires to address to the sensor, we coat or insulate everything with insulating polymer PDMS, which is dimethyl siloxane. And this is how it looks, the sensor looks after it is done. This is actually after it's used, but it is transparent everywhere, except the carbon paint that we were using to address the sensor. Once the sensor is done, we place them onto the participants. So in this left picture, you're seeing the strain sensor in the middle of the throat. And in order to get improve the accuracy of our machine learning later on, and also see the different motion artifacts that do come along during swallow, we did use a secondary sensor, a CMG sensor. And these are commercially purchased. We just wanted to have that as a proof of sanity, like we are seeing the swallow indeed in the strain sensor. And again, I explained this in the previous slide. So the layer by layer, the strain sensor is insulated between the two, or sandwiched between the two insulating PDMS layers. You have a monolayer of graphene with gold nano-islands coated with the conductive polymer that's commercially available, PDOT-PSS. And I have also shared this picture before. This is the electron microscopy image of the gold nano-islands. So the cohort studies, we have done them while the participants were seated in a very relaxed position. So the only motion we were detecting was on their throat when they were swallowing the volume that they were given. We have tested this on 11 participants, mostly grad students, because it was COVID times, so we couldn't really recruit a lot of people to come in. We have used a commercial data acquisition board for this, which is connected to the laptop over here via USB. We have given them different volumes of water, and we repeated each volume 10 times because we did need more training data for the algorithm training. And we have asked participants to actually hold the water in their mouth. So it's the first stage that I introduced to you in the 4-stage model. Hold the water in their mouth for 10 seconds. Then they were instructed to swallow, and then hold for another 10 seconds to get the baseline signal when they're not swallowing. And this is how the data for strain and SCMG raw data looks like. So here you're seeing the resistance change from the strain in around 10 seconds or 11 seconds when they were instructed to swallow. You see a peak appear for both of the sensors. However, you cannot just use the raw signal because there's a lot of motion artifacts that does get added into this. So you have to do signal processing. After signal processing, we identified the swallow by finding the first or highest derivative in the data. And then comes the magic part, which I got to collaborate with Professor C.K. Chang's lab, feature selection from the signal for machine learning algorithm. So for this, Poya, she graduated, she was a grad student in Professor Chang's lab. She has actually done this algorithm where she picked 4 fiducial points on the signal, strain signal. And if you look at this top right plot, you see the 4 points being 2 of them being the peaks, the 2 peaks that appear during a swallow, which we have been observing, and 2 valley points that also appear around the swallow. And she basically was taking the difference between the time difference between these peaks or these points, and also the intensities or the height of these peaks as well. And there were more features that were selected, and I can go into that detail after the presentation. But essentially, after picking these features from each signal, she did a 3-fold cross-validation testing of the algorithm. So every participant swallowed essentially 60 times. She divided those swallows into 3. So each little box you're seeing here is a total of 20 swallows. She kept 40 of them as the training data, and the last 20 of them as the testing data. So the algorithm was being trained on 40 swallows and being tested on the last 20 swallows. And you do that 3 times through, and then look at the prediction results. And here is what we have gotten. So looking at the volumes, the sum of testing volumes over accuracy. So the machine learning algorithm was able to predict the volume that was swallowed more than 90% with more than 90% accuracy, which is quite good. However, we did see that the middle volumes were predicted a lot better than the 5 ml volume. And from our observations, we have actually witnessed that the 5 ml volume was a little bit harder for people to swallow consistent every single time, since it was almost being mistaken as the saliva. So we got a lot more error occur on that one. However, we have pretty good accuracy on the larger volumes. But we were able to detect every single volume differently, and we're able to predict that with the algorithm. However, there's still improvements to make. So one of the first ones is that the predictions can be improved with more training data. And this is what we actually get from this first plot. When there is more training data, the algorithm can be trained a lot better with every scenario, and thus you can have better predictions at the end. And we have also found that the sensor platform needs to work for real conditions or real life conditions. We have had participants seated, very relaxed, not moving at all anywhere, and just swallow. So you're minimizing those motion artifacts that could happen at home if they're using it at home, not appear in the controlled lab environment. So this brings me to my next project, which is actually testing this sensor platform while people are in dynamic conditions, meaning that they're not just seated and relaxed, they're either walking or biking. Especially for this experiment, you cannot be attached to the desktop. So you do need to have a wireless data acquisition, which is what we have worked on extensively in this part of the project. And again, we have done swallow experiments just like before with water, different volumes of water, and use the algorithm to predict these volumes this time through. And the first improvement, like I said, is using a wireless data acquisition board. So we can move away from the desktop and have people get tested on field, essentially, and not connected to the computer. So for this part, we have collaborated with Professor Mercier's lab. Vineal, a master student, has helped us custom design a printed circuit board, PCB, that had its own battery and wireless charging coil. So we were not attached to any wires to a laptop or a battery source or energy source. And it had a two-way communication which uses a BLE, Bluetooth Low Energy, which was transferring the data to a phone app where we were collecting all the data while we were having people swallow these different volumes. This is how the setup looked like. So we had, again, our conventional SEMG electrodes and our strain sensor that we make in lab, now with a wireless board. The first experiments, set of experiments we did offsite was actually at Gatorade Sports Science Institute in Chicago. Here we have tested people on a stationary bike or on a treadmill walking. We have tested 7 people here and used our wireless board for the first time, and it actually worked quite good. We have tested volumes again, 10 to 30. These are representative plots that I'm plotting just to show you how the data, raw data looked like. So in red, you see the SEMG conventional electrode picking up the swallow around 8 seconds here, and the blue is showing the strain signal appear for the swallow for 10 ml of water while the person is biking. And while the person is walking, you see the same EMG lining up with the strain around the same time when the swallow happened. So a few observations we got from this offsite experiment was actually really not about the strain sensor performance at all. It was about the SEMG. The durability and the conformability of these conventional electrodes were really not that great. So the conventional electrodes, they are silver chloride, and there is a wet conductive gel. It's between the silver chloride electrode and the skin. However, it was too bulky in general. As you see that not everyone's throat area, that area is very large. So we weren't able to place them right on the muscle whenever we wanted. And secondly, these did not really stay on well on participants when they started sweating a lot. And when the biking conditions, for example, we have people sweat quite a lot, and these adhesives were coming off very easily. And then we were getting distorted EMG signals. In addition, the gel electrodes are not made for long-term use. So that gel, wet conductive gel that's between the silver chloride and the skin does dry out over time. And when it dries out, you're not getting good signal to noise ratio for the SEMG. The second thing we did notice was that the motion artifacts from the cables. So I haven't really seen that coming, but I'm glad we have done this and saw that the cable movements for strain or the EMG, but mostly for EMG, was picking up a lot more noise in the signal. And this is due to these electrode is moving on the skin, and whenever there is any movement, you're losing contact with the skin and getting the noise in your data. So a few improvements that we decided to have is having more conformal and flexible SEMG sensor to have that better contact with the skin and have it last over time, even when people are sweating. And secondly, we have, I have found in literature the dry electrodes are a lot better than gel electrodes. So is it possible to make dry electrodes in lab? And thirdly, removing these movement artifacts from the cables by changing this setup a little bit more. And the solution to dry electrodes is actually conductive polymers. And conductive polymers have been used in many fields, not just SEMG. And one of the most commonly used one is polyethylene dioxythiophene polystyrene sulfonate, which I've introduced before, PEDOT-PSS. This polymer has many advantages, but the most important one for the SEMG purposes is that it has combined electronic and ionic conductivity. In addition to that, it is biocompatible, and its processability is great, meaning that you can dissolve this polymer in water and be able to spin coat, spray coat, dip coat, and every fabrication step, it is possible to use it very easily. And it is also thermally stable. It's quite stable over changes of temperature, but it's also, because of this, it is commercially available, and this is what we have been using before. However, there's a few disadvantages to this. I know it sounds all great, but it is not really stretchable. So you can only stretch it up to maximum 10% strain, as it was indicated in the literature, but that is not great for a wearable sensor. It is very brittle. And there's usually these small molecule additives that people use to mix the PEDOT-PSS with to make it more conductive and more stretchable. This is all great and it's been done before. However, these additives do leach out over time, causing sensor failure and even toxicity to the person. So we wanted to move away from doing these additives into the PEDOT-PSS. So previously in our lab, our postdoc, Dr. Rachel Blau, has come up with a formulation or synthesis of PEDOT-PSS block copolymer with PEGMIA. So in her synthesis, she actually was able to get a block copolymer consistent of the PEDOT block of PSS appended in varying blocks of PEGMIA. So when you extend the changes of polymer PEGMIA into the PSS, you get a lot more stretchable polymer. And when you add the PEDOT to this mix, you do get a conductivity increase overall. And for this process, she didn't need to add any additives because this polymer was synthesized in as one, so she didn't have to add any small additives to make it more conductive or stretchable. And she found that 1:6 ratio, so 1 PEDOT-PSS molecule to 6 PEGMIA ratio was found to be the most stretchable and durable formulation in her library that she has done. And in comparison, you see a stress strain curve of these polymers tested. So the black one is the commercially available Clevios PEDOT-PSS. And as you can see, around like less than 10%, 5% strain, it is failing and it's fracturing as it's getting strained to that. However, in block 6, you're seeing that it can be stretched all the way up to 80 to 70%, which is quite high and needed for wearable sensor purposes. And we have also found that this block 6, when made it into a thin film, was a lot more conformable with the skin. So it was attaching or conforming better with the skin, meaning that it can make better contact with the skin for EMG purposes. And I will refer to block PEDOT-PSS PEGMIA block copolymer 1:6 ratio as block 6 because that's what we called it in our lab in the library. So how did I make sensors out of this? Rachel was kind enough to give me a batch that she synthesized, and I would take this polymer and pour it over a drop cast that was made with 3D printer. So this mold was around 2 centimeter by 2 centimeter in square shape. I would drop cast about 1 milliliter of block 6 in it and cure it on a hot plate at 60 degrees Celsius for 2.5 hours, and then peel off this film and address it with a wire. And the dimensions of this square was 1.7 centimeter to 1.7, and the thickness was around 41 micrometers. And so our experiments, we decided to have Tegaderm over both sensors after we attach them to the skin to keep them more sticky. Before we can use this, though, we need to understand the electrical properties and mechanical properties of block 6. The first one is that we have done an impedance spectra of the block 6 and in comparison with the commercially available PDOT-PSS, which is Clivios, and silver chloride, the conventional EMG that I was using before. So in this impedance spectra, we have done a sweep over different frequencies and measured the impedance over 60 minutes. So the yellow one is showing the commercial PDOT-PSS, and you're seeing the blue one is block 6, which is lower in impedance, meaning that it is better for detecting or having higher signal to noise ratio on skin. And you're seeing the best one being the conventional silver chloride. And over time, you're seeing the time goes up to 60 minutes, which is how long our experiments were taking. And the impedance did not change over time, which is what we wanted to see in these sensors. You don't want the impedance or electrical properties to be varying so much with sweat. Otherwise, you are going to get different signal over time, every time you're measuring. And just for our reference purposes, the EMG occurs between 10 to all the way up to 200 hertz. So I am reporting the numbers for 10 hertz and 100 hertz in comparison with the other commercial material that are used. The next step is checking the mechanical properties. So for this, we took our block 6 thin films, we stretched them until they failed. You have seen this plot before. We were able to achieve up to 80% strain. So when this film is stretched up to 80% of its initial length, it was able to withstand and then break after that. Whereas Clivios, the commercial PDOT-PSS, did not. The elastic modulus, or the softness of this polymer, was close to the skin modulus on the neck, which was around 2 megapascals, and for our block polymer it was around 16. So knowing this, we wanted to do a cyclic load test. So we wanted to make sure that the sensor can work being stretched and relaxed for long periods of time over and over again. And we have done that over 5% strain, 10%, 15%, and 20%. And we have done this over 400 cycles. So we did that for 400 cycles for each one of these strains. And the linear range for our block polymer is around here, so up to 10%. So we do expect to see up to 10% strain. We shouldn't see any change in or very minimal change in the electrical properties, and you might see some for 15 to 20. And you see in these plots, the 5% strain over time, over 400 cycles, showed very minimal change, almost none to its electrical properties, even though it was being stretched and relaxed. And similarly for 10, 15, and 20, there was a slight bit of offset. However, it wasn't drastic. As some sensors do. So after knowing both of these, we have gone ahead and done cohort studies with 10 people. We again used our wireless board for data collection, tested 10 to 30 milliliters of volume. And you're seeing here on the left plot, we have people walking while they're swallowing the volume. And on the right side, while they were biking, they were swallowing that 10 ml volume. And in both cases, you're clearly seeing around 9 to 10 seconds, the swallow peak appear for EMG and also for the strain around the same time, which was really promising. So we have done this for walking and biking. And I, as a triathlete, I'm asking, why can't we do this while swimming? That was a very bad delivered joke, but we'll go with it. So as you see from the previous plots, it was very clean and nice looking signal, but it does not start that way. You do have to do signal processing and filtering to get to that point, especially for dynamic conditions when you have a lot of moving parts in your system. So here I'm going to talk about a little bit of the filtering that we have done. So for the walking on this left plot, you're seeing the raw EMG data, which does have this DC offset in the potential that is measured. And when you look at the biking, again, you're seeing a lot more DC offset caused by the movements of the body. However, there's ways around getting rid of this noise that is not wanted. And the first one is the electromagnetic noise, also referred as the power line interference, that arises from the radiation from all the power sources that surround us. And that's around 60 hertz, and that's known. So we can apply a notch filter to get rid of that first and foremost. And the other one is the internal noise caused by the subcutaneous tissue between the electrode and the active muscle that we're trying to measure. And that can also partially be reduced with high pass filter. And we've used a 30 hertz cutoff frequency for that. Similarly for strain, we have used a band pass filter with 0.5 hertz as the low and 10 hertz as the high cutoff frequency. And you do have to apply a smoothing filter because, as you can see, the motion artifacts do appear for both of these cases. And we use a very well-known Sawicki-Golay filter that's in third order. And also a moving window of average that is also applied. And I have to mention that this part was also in collaboration with the Professor Cheng's lab with a different PhD student, Koen Jung, who was helping me with the data processing and the machine learning of this project. The next step was, again, feature selection. So we have seen here, as described before, we have the EMG sensor where we detect the swallow peak appear for both strain and EMG and use certain features to be used for the algorithm. The first one is the SEMG summation. So that's the summation of these intensity peaks that do appear during the swallow in the signal. And the next one is the SEMG width, meaning the time it takes for the swallow to happen on the SEMG signal. And the low frequency power. So the power that is like the root mean square that is calculated in the low frequency regime for the EMG signal. And for the strain signal, again, we see the two peaks appear. And the first one was measuring the distance between, or the time between these two peaks, which is peak to peak width. And the next one was the peak skewness, meaning the difference between this valley point to the second point, or time it takes between those two, minus time it takes between the first point and the valley point. And the last one we've used is the peak offset between the EMG signal and the strain signal. So using these two, we have gotten prediction results. So for this, you're seeing the blue is for the walking, total walking predictions, and for total biking is in orange. So this is for all the participants combined for prediction. And on the x-axis, you're seeing all the volumes and the prediction error on the y-axis. So on average, we have seen the walking experiments had better prediction performance than biking. And if you remember, we did see a lot more motion artifacts appear, so that was to be sort of expected. And we have also seen the middle volumes were predicted better than the extreme volumes. There are again improvements, and the first one is testing different models for machine learning. So I have learned a lot about machine learning in this project while I was doing with our collaborators, and essentially they have informed me that there are different models that you can try, different features that you can try in combination to be able to get better prediction results. And again, we have again and again have come to this conclusion that we do need to increase the amount of data for training. However, this is really hard because everyone is swallowing 60 times, and that approximately totals to 1 liter of water that one person has to drink just to get enough data for this kind of prediction. And that is a lot. Let me tell you, people have to go to the bathroom quite often with the sensors on their neck. So we do need to, instead of increasing the number of repetitions, we do need to get more sensors on the patch to get more data out of one swallow. And that brings me to the last chapter, which is pattern strain sensors. And this is because we want to collect more data per swallow with multiplexed version of the strain sensor patch. So instead of one, you get multiple sensors on the same area. Secondly, we do want to obtain special strain information. That's possible if you have an arrayed sensor in the patch, so you can get strain information from different areas during a swallow. So that's also valuable information that we can use. So I'm going to introduce the first version of the multiplex strain sensor. And as you're seeing in this picture, it's the same fabrication process, but instead of having one strain sensor, I have 8, and I've labeled them from 1 to 8 over here. And they are the same process. However, these are each addressed individually, meaning that I was measuring the strain signal individually from each one of these sensors. I've tested with saliva only, so I had asked a participant to just swallow, dry swallow, no water. And in all of the 8 sensors, you can clearly see the swallow peak appear over noise. So that was very promising. So I got excited and I gave them 10 ml of water, and that also appeared very clearly for 8 of the sensors. And lastly, I've tried hard bolus like a cracker. So after chewing the cracker in their mouth, they swallowed. And again, we have seen from all sensors the swallow peak. However, this wasn't, this was just a proof of concept, and I had to, have the person swallow 8 times for getting 8 signals from him. So version 2 is actually improving this data acquisition in a closed loop circuit way. So Laura Becerra, a grad student from our lab, helped me design this setup because I am not an electrical engineer. So she has come up with this design where we treat the strain sensors as resistances in series. And when you apply a constant current that flies or passes through each one of these resistance, you can measure the voltage difference across each sensor and then calculate your resistance that way, because we know that voltage is equal to current times resistance. And we have done that using our strain sensor. So we have bought a commercial differential amplifier that can measure the potential difference across the resistance. And we have used a current source to apply the constant current that passes through each one of these sensors one by one and comes out from the other end. The fabrication for this was quite similar to the previous one, where we have a mask that when we were evaporating the gold nano-islands, we used a mask to only evaporate in the areas that we wanted to. And after that, we had added the gold film to connect these strain sensors to each other, so they're in series, and transfer this sensor film onto a tattoo paper so that it can be put onto skin easily. And this section is how it's different from the previous fabrications, where we were able to do masking method to only put the sensor in the areas that we want. And this is how it looked. The sensor looked on a tattoo paper. So you seeing the gray or blackish areas is where the strain sensors are. This gold area you're seeing is an actual gold that's connecting all the sensors to each other. And then we get the voltage readout across each sensor using the wires attached. So our first experiment on the throat was sort of successful. So we did see swallow peak appear for 10 ml water for each one of these. However, for some of the sensors, we did pick up some of the line noise that was appearing. Even though we were able to detect different swallows, different volumes of water for each one of them, we still saw these appear in every single trial, the noise appear every single trial. And there are solutions to this. So our next step is actually to use polyimide, which is another polymer, insulating polymer, to use as a support layer for gold. So gold is not very stretchable when you evaporate it and transfer it onto a flexible or stretchable substrate. Gold metal film is not going to stretch with that elastomer. The adhesion is not that great. So you can overcome that by adding polyimide and then evaporating the gold on top. Then it is more stretchable. So our next version is including that polyimide layer and the fabrication before we transfer everything onto our PDMS sandwich layer. And in addition, we are using these copper wires to address each one of the sensors. And that is not very ideal. So that was our solution at the time. However, we are moving towards using an anisotropic conductive band, which is essentially going to attach or bond over here. So you're going to get rid of all of these wires that are coming out of the person's throat and just have it come through this one band that is also insulated and commercially available. So in conclusion, we demonstrated for the first project that detection of different swallowed volumes was possible when people were, in a stationary condition, and we were able to predict these volumes quite well using a machine learning algorithm. And then we moved on and tested this setup in a non-stationary condition. So people were moving around, and hopefully this was more close to what people will be doing if they're going to test the sensor platform at home. And in that case, we were also able to detect the swallow, which was our first goal, and also able to do predictions with it. And lastly, we have demonstrated that proof of concept of pattern strain sensor, so we can get more training data to have better understanding and better algorithm results from our studies. And in addition to that, we can use this pattern sensor in a different study where we can figure out the spatial strain mapping of the skin during a swallow. And lastly, I will talk about my non-thesis project. So this project I have worked on my first 2 years of PhD in Professor Donald Serbely's lab. And here I have worked on making a compact and ultra-sensitive nanofiber optic force transducers. And this sensor is essentially uses waveguiding nanofiber called tin dioxide. We would synthesize this in lab, functionalize it with a polymer graft, and add an optical signal, which is the gold plasmonic nanoparticle that can give the light out in the far field. And we were able to measure forces that are applied on this gold using a camera on the far field. And this project was actually really cool. We were able to use for example, detecting a bacteria swimming force in the solution when this nanofiber with gold nanoparticles are in the same environment, and as well as detecting cell beating frequency. And again, this also acted as an acoustic force sensor as well. And we have actually published the methods and materials in very much detail in Nature Protocols in my first 2 years with my coworker, Dr. Yusong Shi. So here is a list of my publications over the years that I was here at UCSD for PhD. My last 2 papers are in progress, so we will hopefully be done with those very soon with the last revisions. So lastly, I want to thank everybody who has helped, especially my committee members who are here today and agreed to be on my committee and give me quite useful information, quite useful comments during my Senate. I like to thank all my collaborators for machine learning part, because I did not know that myself, but I learned quite a lot about it, which was really exciting. And also the custom PCB board. I'm, again, not an electrical engineer, so I really needed help in designing these, and move away from desktop was really cool project to work with. Our collaborators in PepsiCo, so Dr. Lindsay Baker and Dr. Michelle King, who were great hosts for us and actually very kind to invite us to their site to do this offsite experiment with our sensor platform. And lastly, all my Lipomi lab members, lab mates, they have really made all these projects happen. I have not worked alone this whole time, and it wouldn't be possible to do all this work alone, to be honest. So you really need good collaborative work environment to get all of the important and exciting work happen. And also my Lipomi lab members that are not currently in lab, but they have graduated, Dr. Julian Ramirez, Professor Laura Kaiser, Professor Charles Dong, and all the other previous members who have helped me in first 2 years. I have to thank Professor Serbely for getting me into his lab when I first applied, and Dr. Yusong Shi and Dr. Connor Riley, who were great mentors and collaborators for my first 2 years. And with that, I will take any questions now.
Okay, people in the audience can ask questions live, and if you're on Zoom, put them in the chat and Rory will read them.
I can actually see them here too.
So I guess you see a potential application for this of like diagnosing
So yes, but that the sensor needs to be, or sensor platform needs to be trained with none of that. So if you're talking about dry swallow, if they just need to swallow once, yes, that is possible, but we do need to train the platform for dry swallow, which is just a saliva swallow. So that is possible in the future, but again, needs more training data for, being able to predict that kind of signal as well. Yes, Rory.
So when you were showing the array sensor,
Yeah.
It seemed like when you were measuring it individually, it seemed like actually like 7 of them looked very similar and one of them maybe looked a little different, which isn't surprising. If you're scaling it down, it makes sense. You might have some that don't work. So I guess my question is, do you anticipate having the algorithm know like to discard like bad data essentially, or?
Right, so that goes into the error compensation. So another reason that having array sensor is beneficial is if one of the sensors does fail for some reason, you have 7 other sensors that you can train your algorithm with, which is an added bonus to having an array sensor. But one of the reasons why they all look a little different is because of where they were placed on the throat. So some of them were closer to the middle of the throat and some of them are farther away. So you're going to see slightly different strain applied onto each one of these sensors, and thus they're not all going to be looking the same, which is good because you need all of those different scenarios. If they were placed in a wrong place, or everyone's going to put them on their throat in a slightly different orientation. So that is good to have multiple sensors where algorithm knows, okay, I have to check all 8, do the offset of each peak, and then correct the errors of putting in different locations.
You have some questions.
Which one is the first? Samar asks, how does the half wave peak width of motion artifacts and penting differ from that of swallow? Okay, so that is a great question. So all of those moving artifacts that do happen other than the swallow, EMG swallow peak are happening at a different frequency range. And therefore we can actually get rid of those motion artifacts when we apply those high and low pass filters that we have applied in the EMG and also for the strain. So they are existent. If you don't do any signal processing to your data, they are going to appear. However, they're in a different frequency range, so you could filter those with specific frequency cutoffs. But good question, Samar. Oh, Paul.
Thank you for the talk, Pete.
Thank you.
What kind of predictive machine learning algorithms are you using, and are they resilient to small training data?
Yes, so the first one, so the question was what kind of predicting models we were using for the algorithm and if they work good for a small number of data sets. Is that correct, Paul? So yes, so for the first one, we have used a canonical correlation analysis. And if you ask me what that is, I can only give you very surface level information on that. And for that, we have used for the first part where we didn't have a lot of motion artifacts and we did have quite low data, but we were able to get better prediction results. For however, second version, the second project, we could not use the canonical correlation because of the motion artifacts. We were actually getting worse prediction results using that model. So we moved to linear vector support model. I'm hoping that you know that, but so essentially I was told that it was better for when you have more motion artifacts in your data, when there's movement artifacts more present in your data. So for that second part, we have to use a different model. So my cousin Selan actually is asking a question on Zoom. If you would have the possibility to work with a really soft material for the sensor, which could stretch a lot, how would you avoid noise in your signal? That's a good question. So are you asking like the softness of the material is matching to that of skin? Because we have only 1 magnitude higher elastic modulus to the skin with our sensor, which is quite close to that of the skin itself. So in that way, we did see noise, and it's not really possible to get rid of all of the noise, but you can reduce it, and you can reduce it with, again, the filters, making sure your cables are not moving around or touching the skin. You make direct connection to your cables, the wires. So there are ways to do it with filtering, and there are also ways, making it stick to your skin and adhere to your skin a lot better. So those are a few ways that you can decrease the noise.
One last question.
Yes, Ben Sarno.
Other than the complexity of the experimentation, Was there any particular reason that you didn't run underwater trial?
So Ben is asking he's referring to my terrible joke that I tried to make, which was, can we use this on while people are swimming? And that would be only possible if we could get the board, PCB board, to be water resistant in one way with casing of some sort. And also you do need to change the copper wires to something that is insulated, a lot more insulated than what they are right now. The sensors would last because we have Tegaderm, which is water resistant. However, the board is not. So it is possible to do if you can make the board encapsulated in a box that is not going to leak any water in, just like the cameras, underwater cameras.
Can you imagine what the water interface?
For the cables, yes, because the cables are going to move around. So you need to be able to make sure that are adhered to a skin, like insulated and touch your skin really well. So you're not going to be swimming those. But again, that type of noise can be filtered out because it's happening in a different frequency range. As long as it's not in the 30 to 100 hertz range, we are essentially good.
Very nice.
Thank you.
Okay. Let's thank B again. Okay, at this point,